So it's a real pleasure today to start off the day really focused with an overview of research. I know you have had an extremely busy 2 days, the first day with a lot of information, and yesterday I'm sure very exciting visits with your representatives and looking towards getting their support for many of our public policy priorities. So it's a real pleasure today to switch gears a little bit and talk to you about our internal research program. But in doing so, I want to be sure that you know that all these things work closely hand in hand. Some of the achievements through our research program have enabled the programs that are now at DoD, together with your support and the hard work of the teams in the public policy area. So when you think about it and the stories I'll tell today, it all is part of a large team and a partnership. But before I move on today to that, I wanted to just alert you to what's going to happen research-wise today. First of all, to make sure that you are aware that Brian Traynor, who I'm not sure if he's in the room at this particular moment in time, but he's here again today and will be doing blood draws for our important studies in biomarkers and genetics. So encourage you, if you'd like to participate, there's a big sign, it's just outside of the door. If you can't find it, ask one of our staff, they will get you there. The other is to remind you that after this session, we will have clinical trials updates, and I will introduce my colleagues at that time after our break, and then encourage you straight after that, when questions are finished, to enjoy and join us in the Research Expo. There are many other expos, but I'm going to promote our program there. We have 2 young fellows that are going to be there who will really talk to you directly about the research work they're doing. We have our team from Niels who will be there to answer questions about clinical trials, and we have Dr. Scheffner on behalf of Cytokinetics who will talk to you about any of the trial questions there. So encourage you to come. It's very informal. Don't feel intimidated. Any question's a good one, and we welcome that more personal dialogue after the session. So with that, I think we can start. I think the announcements are done. I will start with the session. And I always try to do something new, but in fact, as I walked in the door, Kathy excitedly said, "Boy, there's so many new people here." So I hope that I haven't forgotten to give you all the details you'd like to know, and truthfully, in an hour, it's a very difficult thing to do. So whatever you think you need more clarity on, if there are areas you haven't heard that you're really interested in, I'm always around, I'm here all day, and I'm certainly available through email contact or by phone. So feel free to ask the questions. So today what I'm going to try and do, let me just get everything up and running, and I hope you can all hear me clearly in the back. Maybe eventually we'll close the doors just to be sure there are no distractions. Today I'm going to talk a little bit about the biology of ALS. The bottom line is if we don't understand the disease, it's going to be terribly difficult to find out what kind of causes we should get rid of and what kind of treatments we should try and embark on. So that's really critical, and I'll do a very, very simple few slides on that. I would like to talk about our program. We have, over time, acronymed it TREAT ALS, Translational Research Advancing Therapies for ALS, because actually everything we do and fund in our research program is with that goal, to find treatments and a cure. I will talk about drug discovery, and in particular for ALS. It's in itself a very systematic and specific process. I really want to highlight the importance of model systems. I think there's a lot of misrepresentation about what models are important, disappointments, why models don't really replicate, and why is that, and I want to give a good sense of the field in that. And then I think at this moment in time, one of the key areas to really help enhance treatment development is biomarker development, and so I've got a focus on that today. But so again, a slide that some of you who are not new to this have seen many, many a time, and I do this to illustrate ALS in itself is a complex disease. It's not one region that is affected. It's not a bruise on an arm or one limb that's affected. It's a system that goes awry. And that is really part of the challenge for therapy, and part of the challenge for understanding the disease. But we have made progress, and certainly we know, and we have known for years now, that ALS is a disease that affects the upper motor neurons, which have their origins in the brain, with long projections to various regions, the lower motor neurons in the spinal cord, and all these connections connect into muscles which need to be innervated for function. And so if any of these systems, from the cell body right up here through the processes or the connections with muscles, go awry, the outcome is that you have the challenges with walking, movement, respiration, and what you all know very well as the disease ALS. And when I stepped into the field in 1993, and in fact this was fairly much the representation of the field for several years, ALS was seen as a real problem of the motor neurons, and it is indeed a problem of the motor neurons. Unique nerve cells in the brain and spinal cord that have a very small cell body where all the genetics is made, and a very long projection that connects distances of up to 1 meter in humans to get the messages from the cell to the action that has to be done in the muscle. And so I would say it was a fairly motor neuron-centric time, and we looked at pathology in human disease, we looked in patients, and we saw that that's what a normal motor neuron looks like. It's just the cell body here with the nucleolus and the messages and the genetics here. And in ALS, those very beautiful regular cells were balloon-like, swollen, had lots of collection of things which we call aggregates or accumulations. That's not a good spot with the microphone. But we see these blobs here. And in fact, here as well are the long axons that become shriveled. That was sort of the picture we had at that time, both in the clinical picture and what we knew about the disease in the lab. Actually, what has evolved over the time, and I would attribute that to the finding in 1993 of the first gene mutation, and I'm going to talk about that kind of connection a lot, things really changed. We suddenly were able to experiment and build models in a dish much more effectively to tell us that the disease was indeed motor neuron centric. It's the motor neuron or the nerve that dies and the connection is broken with the muscle, but it's much more complex than that. It's also the milieu around the motor neurons, the astrocytes, that are very important for keeping the health of the motor neuron in check. And I just remind you, this is a beautiful image of a neuron actually from a rat model. This is that cell body, and I promised you a single axon, and that is true. But what is interesting is that there's many, many processes called the dendrites, and they too have to make connections. So it's not a simple one-way connection, it's an interaction of a network of things. And then to the right, a beautiful image from Jeff Lichtman and his group. That which you can't see is the striations of the muscle, and this is the connection of that motor neuron to the muscle. Beautiful connections with boutons that exquisitely connect and send signals. Well, this too in ALS dies back and shrivels up, and so another area that we have to understand better. What causes ALS? Everyone all have their own ideas of what they were exposed to, what they did that they feel was a trigger. Here is what we definitely know. Genes definitely cause ALS. 5 to 10% is familial, where it's carried through the family, where there are more than 2 who have ALS within that family. That's considered familial. Sporadic is those 90% that don't have a direct familial link, so it hasn't gone through the generations. This area has become murky, and certainly if you want to talk more about that, Brian Traynor, who will be here, is the person to talk about it. He's really one of the lead geneticists in this room. There are many others, but he's certainly done tremendous work on that. And he will tell you, as will other geneticists, that it's much more murky, the sporadic, where genes play an incredibly important role in sporadic as well. Whether it's a single gene is unclear. Most likely it's multiple genes that interact to cause the disease. It's a disease of aging, so certainly you will see, and you are familiar with those that are younger that have the disease, but in general it's between 50 and 60 is the general lifetime expectancy of having ALS. So aging and our normal aging and wear and tear of the body is contributing to the disease. There are several risk factors, and I know you've heard a lot about, and what you go and advocate about is the fact that there's a 2-fold increase in veterans. So we know that there is something, but we don't know what factor it is at this point that is causing them to have it more frequently. Smoking is one of a very well-published risk factor. Risk factors are tricky because actually in Guam, where there was a 40-fold increase in ALS, which has now actually disappeared, we still, as clinicians and scientists, debate about what is the cause there. Some believe it was a cycad seed. Some strongly believe there might have been some genetic factors that have now disappeared. So identifying and then resolving how risk factors are involved is a tricky subject. So moving to our programs, and our translational programs have many components. They all work together. Some are smaller projects we fund, some are larger. Some see the opportunity for many millions from our partners through industry and government. And I really draw it, as one would see often, a pipeline for the industry development, and I think there are images in your advocacy materials that look similar, but perhaps in more lay terms. You start at the very beginning to develop drugs by identifying a target. So a lot of our programs are to understand disease mechanism. Once we think we have a target, we really have to be sure it is one, and we have to prove it through testing it in model systems. We then go through processes of therapy development and many clinical studies. I've chosen in my presentation not to talk about all the clinical activities that we support, because you will have a focus on that coming up afterwards. But we certainly play a pivotal role in help seeding studies in clinical arenas, both with those living with ALS today. Our management programs are designed to help improve those living with the disease, as well as clinical trials for hopefully future good treatments and potentially moving forward towards a cure. And the two areas which I will spend quite a bit of time talking about are what we do incredibly well for the community most broadly, is building systems that can facilitate research. These are the model systems, and I will talk extensively about biomarkers because they are so critical to move things forward. And so we do have success stories of where we've identified a target, and through our funding programs have been able to get a study into clinical trials, and I will mention that a little later. This is just a snapshot in time, and it constantly changes. In fact, today I'm here at the end of the day to work with our research committee to approve 15 new studies, which will increase the portfolio by 15 projects and over $2 million in investment. So this is, at a current time, is our funding portfolio. And importantly, it is a global program. And that actually, in fact, my colleague Amelie at NINDS pointed that out, is that NIH really does fund, but very, very few foreign studies. They feel if they can't justify it very carefully, they are unable to do so. We feel that we need to fund wherever there's good science. We don't know where the answers will come from. And in fact, over time, rather than isolated studies globally, we actually have large consortia that we fund globally. And that is the most exciting, bringing good ideas throughout the world in a collaborative team. A few of the successes, I think that our commitment in research funding, which enabled bigger funding from the government in discovery of new genes, has really shifted the field, and I'll give two examples of that. We really help to build resources, as I say, tools and technologies that enable ALS investigators to enter the field, and that's grown exponentially. We were the first organization to enhance the idea of translational therapy. There are others certainly involved in that now in the ALS space, but it was an initiative that we felt strongly about and helped mobilize the first antisense trial for ALS, and that was when we reconfigured our programs. We were also in the field, not us ourselves, but the field was the first to really develop model systems using induced pluripotent stem cell technology. I'll talk about that. And we at the association helped support all the preclinical studies as well as the surgical development for the first stem cell trial in the ALS. And we've also, through our partnership with Niels, and I encourage you to learn more about it at our expo booth, have helped encourage the number of clinical trials that can be done. And I think with all of this and with all our partnerships, one thing we certainly have seen, which is tremendous in the field, is an increased commitment by the industry in ALS research. And some of our programs, I think you'll hear today from two young fellows the importance of the Milton Safinovic postdoctoral fellowship. Our Clinician Scientist Development Award, we've now added a training award because actually clinician scientists are so crucial to our research endeavor. Although our young fellows are largely those that work in the laboratory, our clinical fellows bring laboratory ideas together with the clinicians to help move things into clinical trials. Funding of pilot studies is really quite unique to ourselves, although we now partner with MDA, and you'll hear some of those kind of partnerships. The network we've built up, which includes a clinical research institute, which Rick Bedlack was really instrumental in building up, and our drug discovery contracts, which we first started through the association and actually helped drive the current DoD concept and program that is happening now. And just to see a few faces, you'll see two of them today. Our fellows really are the future of ALS, and it's their commitment and enthusiasm very early in their careers that makes tremendous stride as they move forward. Areas of focus that we have highlighted, and some of these are really compiled, BIND groups, genetics, understanding disease. That's the first part of understanding how to do the science, is really understanding the disease process. Therapy development, we work closely with colleagues, clinicians, scientists, the industry to think how we could be more clever about this. Stem cells for drug development and for therapy. Clinical studies, as I indicated, and biomarkers. And today you'll really hear a little bit about genetics, modeling, therapy development, and biomarkers. So why is ALS genetics so important? And this is where we were. In 1993, it was just a happy coincidence that was when I joined the field, part of the motivation actually to join the field, when I started at Johns Hopkins with Don Cleveland. And this was a discovery of the first mutation, which accounts for 2% of ALS. And I always emphasize this because people say, well, 2%, that's a very small number. What is the rest of the cause, and why focus on that? Well, it was the handle in to starting to work with the disease in a much more clever way and think about it more carefully. And I will tell you that this discovery was the beginning of bringing many, many more scientists into ALS research, and they are crucial to make progress for treatments. It was disappointing that it took actually so long for the next genes. The NEFH that you see there is actually a risk factor, not a strong gene link. In fact, it was one of my first studies hoping that they were actually a strong gene link. But what we see in 2004 were the beginnings of finding other genes. But as you clearly see in the last few years, this field has been quite explosive. And really, each gene identifies potential pathways and a target for treatment, and that's why it is so important. We know that most of these genes are very clearly connected, as I said, through families, but actually, many of these genes are now also being found in sporadic disease. And this is just an image taken from Paul Taylor's work, schematically showing you that that small section in the corner there is really all the familial, and this is the unknown sporadic, but what's creeping into sporadic is many of these genes that are also linked in sporadic disease. And so what we have helped support Amar Chalabi in Europe, and this is a global effort supported by several funding agencies, is to help get all these data on these genes online. And there is now a genetic database that captures all of these genetics. We have, with interesting programs, are able to start to think about how these different genes might interlink with each other. And we also have on this database system all the models and the details of those models that exist for ALS. And so genes have really helped us build model systems. And what is important about model systems? Well, it helps us to understand the disease. As I said to you, we were rather motor-centric about the disease, but what we realized with some very clever manipulations of mouse models, when you started to change the proportion of that mutant gene in different cell types, you started to affect the change in timing of the disease progression, arguing that actually cells that surround the motor neuron are also significantly contributing to the death of the motor neuron. And it helps us, of course, also in timing of events, because we know when we see ALS, people with ALS, their disease has already gone on several years. It's unclear how early it started, and that's what Michael Bennett's studies are trying to do. Can we see markers and see how early it's starting? But the likelihood is it's been going on for several years. I don't know how much more beyond that. And we can't then say what are the early events. In model systems, we can try and tease out and say what are the early events, what are the first things that are happening. We have actually, even through our model systems, been able to identify new genes that when we look at the human disease have found that they are actually present in the human disease. And that's remarkable, and I'll tell you two stories that relate to that. We also use model systems, mouse models, rats are sometimes more helpful, to help develop our technologies, to help to be able to inject things that we now need to do in a different way to just taking a tablet orally by mouth. So it's great for technology development. And target identification, you'll hear this all the time. What does target identification mean? It's a clue as to how we might be able to develop a drug or a treatment for the disease. And then you have a picture like this. And I always enjoy seeing at different presentations where Alzheimer's is presented and Huntington's is presented and Parkinson's, we can almost show a similar picture. We just have to exchange the nerve that is affected. Because in all these diseases, all these systems are going awry. The surrounding cells are implicated in Alzheimer's and Parkinson's as well. In ALS, the glia, the astrocytes that exquisitely mop up and control the levels of glutamate in the surrounds, the muscle cells, the interconnections, these are all very important. A confusing but a clear picture, and in fact we can in the mouse models really start to think about what are the early events and the later events, and every single one of these areas presents to us targets for development. So what models am I talking about? And it can be as simple as yeast, it can be cells in a dish, the mouse models, the rat models, worms, and flies. And this is an article that you'll have in front of you, and Aaron did a wonderful job, I think, of telling a very simple story, and do give it a read, because I did ensure that he didn't make it too technical. But he's doing fabulous work in the very simple baker's yeast. And you say, well, gosh, that's so far away from the human. But he identified in those yeast model systems genes that he then went to look at to see if they were in humans. And indeed, they've been now proven to be risk factors in human ALS. And so it's simple systems that can give us important clues as long as we use the model systems in context with the human disease. You can't isolate the two things from each other. Another story, Paul Taylor's done some tremendous work using the fly model, and certainly he's not the only one. You'll hear today from our young fellow beautiful work that she is doing in the fly model, and her PI has done some great work too. And so the fly model is strikingly, in its genetics, quite similar to humans. 75% of human genes can be identified in that fly. It's more how we translate those genes that complexities arise, but the actual genes themselves are often very similar. And this, again, is work from Paul Taylor, and it's an interesting mutation also linked to ALS. But what I wanted to point out, in a fly model, it's the big eye that you look at. And this is a normal, healthy-looking eye. When you insert the mutation, or at least the wild-type protein, it doesn't have a mutation, it looks quite similar. But when you start to put mutations in, it doesn't take a scientist to tell you that those eyes look terribly wrong. And why that is so interesting is that in fly models, you can quickly look for things that will change that, either in a negative direction, what makes it worse, so what genes can you insert to say that gets worse, and that might tell you something about, in humans, if there are co-founding genes that might increase the speed of the disease, or you might have situations where in some cases the speed of disease is much slower, and so maybe there is a gene that is protective, and this can easily be modeled in that fly, and some really good work has been identified. In zebrafish, you have an organism that is see-through. You can actually structure where the neurons are, and that's here. And then you can look at them over time. You can see if you add drugs or if you breed them and mate them with other fish with genes. And then you can start to look at changes perhaps in how their motor neurons develop and branch. And in fact, Wim Robrecht identified in his very early developmental system a mutation or a change that was typical to these fish when a mutation of SOD1 was introduced, which was also seen in humans. And we know now that this could be a modifier effect in patients and could alter the onset of the disease. So this was the first gene identified, the SOD1 gene, and all those colorful blobs are all the different changes that can happen in the coding of that gene to change the structure of the protein. And very interestingly, and for those who listened into the webinar, in fact, it took some time to identify, but there are a series of dogs that also show a motor neuron disease, and lo and behold, the mutation that they carry is in the SOD1 gene. So Joan gave a wonderful presentation of how we can actually use these models together, and from what we understand in mouse models and now in a much bigger species, the dog, how can we link this all to the disease? And currently, she is funded by us to do a clinical study to see if using an antibody therapy approach will work in these dogs. So both the dog owners are desperate for their treatment paradigm, and we know in a much more robust, larger species, we will find better answers. So we are using both the antisense approaches and the antibody approaches in the dog model, as well as other systems. And then I know you heard an elegant presentation from Dr. Nick Maragakis from Hopkins, and you might have seen the slide, I borrowed it from him, that induced pluripotent stem cells are now really presenting a very exciting tool. Some feel this is the answer to everything. I would caution that I think, as I've tried to explain, all of these things in combination strengthen any research identification. So in isolation, they might not be so helpful, but in combination, incredibly strong. And here you take a skin cell from a person with the disease, you manipulate it and then drive it to form motor neurons, astrocytes, cells that you care about, and these can be incredibly valuable both for drug screening and maybe identifying more specific individual patient therapies. And this is just to recognize the Nobel Prize, which in very quick succession was given to Shimanaka, who made this discovery. And this is also just to say, with these kinds of things, you can really use creative microscope technology. Steve Finkbeiner is now able to look at these cells and these iPS cells over time and see how they're changing when treated with drugs or when looking at different patient properties. So to switch quickly to the drug development process and what is involved in that, I think it's important, because I highlighted these things, to even start thinking about developing a drug, you need a target. And so once you have a target, you can think about maybe designing chemistries that will block that target, that will enhance that target. Once you have the chemistries made, you need your model systems to test the concepts. Does it deliver there? Does it get there? Does it reach the target? That's really done very well through academia or contract organizations, as well as in the industry, but through partnerships. And then at that point, there are obviously FDA requirements to take it through to the next phase into clinical trials. But this really is a reiterative process, because what we learn from the failed clinical trials, we bring back into the whole process to see if we can improve a treatment approach. What is very frustrating in the field in general in neurodegeneration, in ALS and Parkinson's and Huntington's, the cost is phenomenal for drug discovery. This is in 2010, was averaged to be about 1.8 billion for a single drug to be developed, partly because there are so many failures. So the cost of research and development goes up, and our ability to find treatments and find successful treatments is rather flat. And so I think over the years, and I think we play a pivotal role in this, what you've done in the last few days in your public policy efforts are critical in bringing many, many more players into the field. And we really help glue the academic and the industry efforts together and bring both the intellectual network, the financial network, and in often cases, of course, most importantly, the patients to these studies, which enable us to move it through the process faster. And I wouldn't have been able to tell you this in 1993 when I joined the field, but this is just a representation of the many companies that are genuinely interested, have programs in-house, are working with the clinicians and scientists on ALS, and that is really very different to what it had been before. But we do have to be very innovative, because small molecules might not be the only solution. Antisense oligonucleotides are one thing I've talked about. Stem cell therapy, many of you have heard and are very excited about it, many hurdles to overcome, but there are opportunities. Gene therapy is becoming much more seen as a way to go. There was a lot of a hurdle at the time when there was a really serious and fatal effect of gene therapy in a patient, which stopped the field, I think, significantly, but is beginning to become a route that's been looked at again with novel and new approaches. And of course, as I mentioned several times, the iPS cells. So antisense technology you're going to hear a lot about, and I don't think I can do it well enough in a few minutes, but in your Research ALS Today, Richard has put together a really nice article that describes the technology, why it's so exciting, and where we think it will go. But it certainly was very novel, and actually this is really how we changed our programs. When Don Cleveland from an academic lab said he had this crazy idea, Richard Smith, this clinician, thinks he could do something which may not work, but would we take the chance? We did indeed. We funded the pilot study. And this is where the mouse model was actually incredibly important, the mouse and rat models, because one thing we didn't know is if we put this kind of a delivery system into the brain or the fluid around the spinal cord, will it actually spread and get to where it needs to be? If that didn't happen, this whole therapy development would have been dead there right away. But it in fact did. It got to the spinal cord, the motor neurons, everywhere where it needed to be. And ALS was the first of the neurological diseases to try this. But now the company who was partnered to this in the very early phases is developing proteins and programs in Huntington's, in Alzheimer's, in SMA, in myotonic dystrophy. So we were really just the beginnings to seed that, and it was really only a financial investment of $400,000 initially to take that chance. It then later became several million, and the company itself has put substantial amounts into it. And I missed the most important slide. This is it very simplistically. If you've got a gene and it's going to read a message to form the RNA, but the message of RNA has errors in it, we just want to get rid of that error and that message so that, and that's done by that red little strand there, that's your antisense oligonucleotide, which is going to connect and parallel up with that area which carries our mutation that we don't want. That means then we're not going to get the protein, and hypothetically, without bad protein, no disease. That's simplistically it. And we knew we could do this with this SOD1 mutation. That was a lot of the work I did in Don's lab. Basically, if you remove this protein completely from a mouse, the mouse doesn't have motor neuron disease. So getting rid of it wasn't the problem. It had something new, and it's something new that we don't all agree about, but it doesn't matter if we can just get rid of it and shut it down. And so importantly, this did go into clinical trials. This just shows how it gets administered into the fluid around the spinal cord. This was a study that had to be done in primates to make sure that you can actually get it to the neurons and for safety reasons. And the trial was seen to be relatively safe. In fact, there were no major side effects. And so now we move forward again, and this is just to demonstrate this is a fabulous partnership with us, the association, and an industry and academic partner. Now we had to move forward to 2011 when a major discovery was made, actually by Brian Traynor, who you'll be excited to meet if you haven't met him, and I'm still not sure if he's in the audience, and by colleagues and Rosa Rademacher's team, and of course many others. You see all the people on the publication. It takes a real team to make these kinds of discoveries. We'd invested in this area for several years, and it was a very difficult discovery to make. And interestingly, they found this not with modern technologies, but old technologies. But the important thing to note, and in connection with the discussion about antisense oligonucleotides, one of the main thing about this mutation, and this is where it is, this is a gene, it's in a region that doesn't translate RNA to protein, it's in a non-translational area. And what happens is you get an incredibly long expansion of these 6 repeats. Normally you have less than 20 if you don't have the disease, and you can have up to over 1,000 if you do have the disease. So what we then hope to do with the same concept is remove that very large expansion with this technique to block the disease. And in fact, although it took about 10 years before we thought to do that for the SOD1 mutations, it took 1 day to decide that this was the technology to use once that discovery was made. And the company and many other companies actually are now heavily invested in it. ISIS has formed a partnership with Biogen, and they are going to move this into clinical trials as quickly as they possibly can. Without knowing, C9orf72 has that ridiculous name because it just is the position of where the mutation is. We have no idea at this moment in time of its function, although we are starting to learn and get some ideas. Other uses of the mouse model that have been very important is developing technologies such as gene therapy. And this is just to explain that if you're going to inject into the brain, this is the region where you would inject, in the ventricular space. And Zhu Sheng has been doing a wonderful study trying to use a different technology called RNAi, and the most important message of it is to use these kinds of vectors. So it's a gene therapy approach using a virus that no longer causes the viral disease, but is called attenuated, so it now will carry it, so it's a carrier of information, whether it's something to block a protein or something to add into the environment. And his important test, again in the mouse model, was to see if you do that with these various viruses, will it get into the spinal cord? And as beautifully illustrated here, all you need to note, this is the brain and the spinal cord of a mouse. Only AAV9 and AAV10 do that extremely well. This is the spinal cord. And again, in the brain, AAV9 and AAV10 are able to go into the system and be delivered there. So that's what you use a model system for before you go to very expensive trials and realize, gosh, you don't even get what you need to get where it needs to be. So again, really with the theme of why model systems are important and the fact that they're so valuable, why do clinical trials fail? I don't think it's because of the animal model. A lot of blame is being given to the mouse that the mouse is not a good predictor. It is only a mouse, and it needs to be done in combination. And there are many other challenges that we have with clinical trials to think it's just the problem of the model. Are we targeting the right population of people living with the disease? The disease is so different in each individual, from slow progression, fast progression, starts in the limb, starts bulbar, that we can't maybe lump it all together in one disease area. And so the other problem is the drug actually getting to where it needs to get to. We know that there's an exquisite blood-brain barrier that protects our brain from things getting in and infecting. So actually when we're delivering a treatment, is it getting there, and is it engaging with that target that we thought was so important in the disease? And actually the other challenge, which we can try and judge from mouse models, but this is where a model system is very different to a human system, is the disease mechanism or that pathway, if it's the astrocytes or the muscle or the incoming anti-inflammatory cells, is that the most important at the time that we treat the disease? Because if it isn't, then of course that treatment is not going to have a very big effect. And actually, when we start to treat that particular pathway, can we see that something in it has changed? And the reality is, will just one therapy be enough for ALS when so many systems are changing? And that's where I lead to a discussion which I will close, and I think that will be my final session before the questions, is biomarkers. What are biomarkers? Biomarkers are signatures of something happening. They can be measured in many different things, and I'll talk about a few of them, but they can be measured, you'll hear beautifully in the clinical discussions today about how you can measure muscle contractions, the forced vital capacity. It can be a measure in the blood, ideally, but that's the more difficult. It can be in the CSF, the fluid that bathes the brain and the spinal cord. So biomarkers are an indicator of something. The best one to talk about, of course, is blood pressure and heart disease. Very measurable. You know then when you take a tablet and your blood pressure goes down, your tablet has done just what it should be doing. So again, I don't think I need to re-stress it, but why is it so urgent? Many things like diagnosis, but today I really want to focus on the idea that maybe we can actually design trials better by grouping patients differently. But how are we going to do that? But via genetics, via a specific readout in the blood, via a specific activity? How are we going to design our trials to stratify more cleverly? how can we measure more quickly if a trial has worked? And you'll hear again the demonstration of the benefit trial today. It takes a long time though, especially if you're going to have survival as one of your measures, several years in the trial. Can we do it so that within a month or 2 months we can decide that drug definitely doesn't work? We don't have to invest so much in it and we can start something else. Very, very important to be able to do trials better. As I said, and it's a special measure that's done, and the industry obviously is very focused on doing this, but we haven't done it very well in ALS trials. Can we be sure that the target has been engaged by that drug? Can we get a pharmacodynamic marker? So in other words, we said the target was important, we said the drug is important, have the two seen each other at all? And this is then the questions that you can then answer. Did the drug get into the CNS? Did it pass the blood-brain barrier? Did we have enough of the drug to make sure it occupied enough of that target? Did the drug do anything to the biology we thought was important? And is the drug being tested in the right patients? Then if we've done all of those things and can answer that, we can honestly say that trial didn't really work. And that's been our challenge in the field. And so although we like to have this image for drug development, we are thinking more carefully that we actually need to do the same thing with biomarkers. And Biogen is actually committing heavily to this area and thinking that as you develop a target program, you have to also develop your biomarker program so that they go alongside each other, and not once you've got a great program, then you decide, let's make our biomarker to test whether our program is great. And some examples, and I think I just highlight these because I think they have real potential, is the fact that neurofilaments, now neurofilaments are a very important part of how our cell is structured. So from that cell body, you have this very long axonal process and dendrites. To keep that architecture correct, the neurofilaments called N, H, and M, H and L, heavy, light, and medium, just because of their size, are very important structural components. What's really interesting, and these are studies now that have been replicated globally, is that neurofilaments seem to increase in ALS patients. And if you look very simply, you don't have to worry about all the detail, but if you look to the left and you see controls, there's a generally low amount of neurofilaments. This is true for both blood and the cerebrospinal fluid. In ALS, although it's variable, it's generally high. And illustrated slightly differently, you can see it here, where control subjects have a very, over time, doing many measures over time, a very flat trajectory. And here it is higher, certainly much higher than controls. Although the trajectory is flat once it's been measured, what is interesting, and Kevin has done, Boylan has done these studies with Jerry Shaw, he shows that actually it appears, and this has to be further validated, that where there are very high levels of neurofilaments, the prognosis and the rate of progression is very fast. And so this could be a very interesting marker for stratification, because what you could try and do then is to get all the faster progressors into the clinical trial and separate them from all the slower progressors. Just a very specific example that actually is going to be tested and is actually being tested as we speak as a potential, and can be then easily measured as long as we standardize these things well in the blood. So I know that Nick has talked extensively about this, and this is one of his beautiful slides I borrowed. He spoke about this on Wednesday. But how can iPS cells maybe help us with more individualized treatments? It's not going to be one person, one treatment, but groups of people that are similar. And this is a very exciting area. Again, to remind you, you can take these skin cells, you can do manipulations to them, you can move them along the direction of astrocytes, those support cells, you can move them in the direction of motor neurons. Motor neurons are developed more quickly than the astrocytes, but you can program them to do that. And then you can, and then just to really acknowledge, and some of the faces Nick, you have seen, and Dr. Sokovich, you'll see here speaking later. This is really a group effort, and part of it is being funded through us in partnership with NIH to build a consortium to develop these techniques. What Nick and his colleagues have really assembled very beautifully are now cell lines from a variety of different gene mutations, a variety of controls, and a variety with different phenotypes, so short survival, long progression, upper motor neuron only, lower motor neuron only, and to try and tease out, when you start to look at these different cells next to each other, How do they look different? How do they operate differently? And are there unique signatures in each of these that we can exploit when trying to select pools of patients for a clinical trial? This is one example where I think C9, that gene mutation with this enormous expansion, has the most initial potential because we have so many C9 patients. Our funding is helping support registries to collect patient samples from these C9 patients. Michael Benneter is doing a rigid study looking early in these patients, just like he did for SOD1. But what is interesting to note, and again, you don't have to see much other than to recognize that there are red dots here. This is work done by John Ravitz and Clotilde in San Diego, where they took the fibroblasts, not even going to motor neurons or astrocytes, just the skin cells, and looked at the nucleus and found these red dots, exactly what Brian Traynor and Rosa Rademakers had reported in their studies. And this is just to highlight that it's both in the nucleus and outside the nucleus in the cytoplasm. Another beautiful example that your postdoc today will tell you more about, and her team have shown, is that when you take the skin cell and then make them into the motor neuron, they too show these exquisite red dots, typical, not in controls, not in those that are taken from patients who don't have the disease, but certainly from patients that do have the disease. This could be a very interesting signature, because if you then treat the person with a treatment that we think works, like the antisense, does that profile change and look more like the human control? And this is again to extend on that. We think that what this big extension does is not only form these clumps in the nucleus, but begin to form a protein, interestingly, a little bit contrary to what we learned in molecular biology in the early days. It can find a site on that big repeat to start making a protein, and that protein, which Leonard Petruccelli has now developed antibodies again, can also be found in these iPS neurons. And in fact, if we look in the brain and the CSF for markers of those proteins, you see in the cases where the C9 mutation doesn't exist, you can't identify it. But where there is a mutation in C9, you can start to see it in the brain and the spinal fluid. Now, this is unpublished data, but has been presented at meetings, and it obviously needs to be verified. But that already is a very, very nice finding if we can repeat that and find a blood test that will tell us and predict that there is a toxic result, and then, of course, see what treatment does to that. Again, much more complicated, but important information we can use. From these cells, we can get these very complicated profiles and see how they are unique to a patient versus someone who doesn't have the mutation. Finally, another area that's really important, and in fact was highlighted by the work of Dr. Stan Appel many, many years ago, and has become a really invigorated theme again in the field, is the role of inflammation in ALS. And certainly it's not like an MS inflammation, but there are now many clinical trials. The TDI, who I know, Bob, I think is in the audience, they're also doing an exciting study in Galenia, early days still, to see if modulating this system is an important one. And this is work of Oleg Butovsky and Howard Weiner. In fact, Howard Weiner comes from the MS field, and so some of his ideas and his skills are being applied. The idea is that if you bruise yourself or you damage yourself, there's a swelling and an inflammation that happens. In the brain, the same thing happens. There is a response to that. There's a good response that's helping repair, and there's a bad response that's actually causing a cascade of events that is now making it toxic. And so green is good and red is bad. And Howard and Oleg and his team identified that microRNA, MER155, seemed to be a potential regulator of that. So looking at these complicated pathways, this was done first of all in a mouse model carrying the SOD1 mutation. And of course, it's only a model, so it wouldn't be of any value if it wasn't happening in the human disease. But actually, when he looks in the blood system and the spinal cord, and what you might not be able to see clearly, but just look at this here. In the human controls, this particular MER155 is completely flat, and in ALS patients, variable, but certainly elevated. So what his team are doing is they're obviously verifying this, and Dr. Berry and others at MGH are working together with them. And then they are developing a therapy to block MER155. Now, for the clinical trial, you're certainly not going to take a population of ALS patients that already have a readout showing that miR-155 isn't elevated. They will then look in patients by doing a blood test to see which are the patients that have high miR-155 levels, and they will be in the trial. So that's just beginning to think about how you are going to select certain populations for clinical trials. You're only going to block a target that is important in a disease, and maybe that target varies from one person with ALS to another. So I think I've really emphasized it, but our strategic directions for our internal programs at the association are really to help identify biomarkers that are going to be effective for stratification. We want to help the clinicians and the scientists better design the clinical trials. And in fact, as in a partnership that we'll probably learn a little bit more about with MGH at NCRI, we were able to fund a study giving them more funds for that clinical pilot study because they had a biomarker development plan to run alongside it. So we don't want to encourage trials that are not able at the end of the day to tell us whether or not they have worked. And finally, I believe that ALS research is all about our partners, and I really think that the industry plays a pivotal role in this, and that their enthusiasm and commitment is increasing is tremendous, and it's through those partnerships and through initiatives such as one we're trying to spearhead with biomarkers, where we can get investment from industry to help try and move the biomarker development forward, which will benefit their trials and their drug development studies too, would be very crucial. So that's really what I intend to help spearhead. A reminder, if you haven't heard enough of research and of me today, we have great scientists that come and clinicians that help us monthly with a research webinar. And we're delighted that in the theme of today's focus on biomarkers, Dr. Lal Ostro will present next month on a biomarker project. We really encourage you to participate. I know you have busy schedules, but it's a half an hour with the scientist presenting, and then a half an hour for Q&A. And your questions and your discussion is really so pivotal to this, because often it really makes us think of things differently. And so that's a really great opportunity to have that kind of dialogue. You'll hear more about this today. I do encourage you, come to Expo. You'll meet our clinical trials liaison who will be there to tell you more about trials, to put a face to sometimes a very challenging area of understanding what's out there. And finally, I'd like to end by saying all the thank yous. It wouldn't happen if it wasn't for all of you in this room, because we wouldn't have to fight to find the treatment and the cure. Thank you for all of you and your families and your caregivers for always being there, for being on the Hill, for being the voice for ALS, because that does make a huge difference. Thank you to our donors. Thank you to all our chapters that work locally to push for research. We try actively to work with our chapters to fund exciting research in their area and engage those scientists locally. To my colleagues, Mark Yard, for being there to run the programs, for Richard Robinson, who I think does a great job at trying to communicate the research to you, and to the whole of the national team who are partners to all of this in all the different divisions. And these are just lists and lists of people that without them, we couldn't have outstanding research programs. These are our advisors, our reviewers, our colleagues, and certainly our partners in government, the NIH, the CDC, the Packard Center for Research, MDA, Therapy Alliance, MNDA UK, many, many other partners who have been pivotal in helping us develop and grow our programs. So thank you for your time, and I have plenty of time before your coffee break for questions. You might sometimes have to wave a hand, because when you're here, you see lights. There we go.
I'm from Lexington, Kentucky, and my husband passed away in January of this year. One of the things that I observed in our support group in Lexington is within an 18-month period, 5 of our husbands died, and it was all different paths, completely different paths. My husband, his death was truly the most horrific. I didn't think it could ever get so bad, never heard it could get so bad. He was a vet, and one of the things I discovered is when I made some phone calls after his death, even within the VA, no one is reporting his case. No one is reviewing his data. And I asked the others, do you know of anyone reporting or reviewing your husband's data? And they said no, and especially with the VA, I just felt like his story died. And with all the research that is going on in many, many ways, I just wonder, all the people in this room with ALS, will their stories die with them?
So I think, and I hope you heard on Wednesday, but they will be here today as well, that's really why the ALS Association was so behind building a registry for ALS. And so there is the National Registry. Absolutely, it doesn't collect historical data, because that's more tricky to evaluate, but it does take all the data from families living with the disease now, and there are the beginnings, and if you didn't hear about it, please do go to see them today at the research the arena and the expo arena, because that is critical. Understanding where someone lived, what exposures they might have been exposed to, their clinical signs. They've added the clinical data that you can see how it progresses individually. So that's the goal of the National Registry, and that's why many of you on the Hill spoke towards building that.
I would like to comment that I did talk to them, and I do understand that, but in the case of my husband, where I didn't have, you know, his log on, and when he finally just checked out psychologically, just didn't want to do anymore, it stopped reporting, and as a spouse, there's no way for me to get in and finish the story. So I think that bit of information, and however it can be cataloged under those circumstances, I think that's important to develop in the future as well. Thank you.
Very good point, and hopefully that's something that can be integrated. Yes. Oh.
Hi. So for the SOD1 gene target that you were referring to, I was just a little bit confused on the target of that. Are you trying to get rid of the protein in its entirety, or just target the mutated parts of the gene? And also, is the specific function in the pathways that lead to ALS of this gene known and understood yet?
So in SOD1, what is being done with the antisense approach is to downregulate the whole protein. It doesn't matter where the gene is. In reality, the system will never shut it down completely. There's a certain shutdown level, so there'll still be some of the protein, but at much lower levels, and it isn't going to choose whether it's an A4V mutation or a D90A mutation, so it's agnostic to the mutation. Good question. The other comment is what is known about SOD1 in terms of mechanism. Well, I think a lot, and I mean a lot of my work, and actually in Don's lab, was focused on that. We looked at pathways of how things were going awry. Certainly SOD clumps up and accumulates, and that's a real problem. Like in anything that accumulates, the system then can't go through normal processing. So you have too much of it building up, the cell's not removing it, and then it trickles down to all kinds of damage. It seems to affect the mitochondria, that's an energy system in the house, in the cell. So it's doing a lot of things, but the logic between shutting down the protein is that if it's doing multiple things, it might take multiple intrusions to making a difference, and rather go to the source. In this case, you can do that. In other cases, you might not want to do that because it's too important and integral into the cell system, so you can't do that. But here, where you have something that we've proven now through model systems, that shutting it down to a large level isn't a problem, and it's been done in humans now, it's safe, this could be a benefit. Does that answer your question?
Yes, and is this approach going to be taken as a preventative measure if you know you have the mutation, or is it more of to improve the symptoms of people who already have been diagnosed with ALS?
So that's also a very good question, and of course it's what always comes up with families that have SOD1, and that's why Dr. Michael Beneter is working on this hard. The question is, number one, any treatment has some risks and side effects, especially we don't know if you keep on taking an antisense therapy if there would be long-term effects. The other unknown is, in an SOD family that hasn't yet shown signs of the disease, how many years before are we going to go in and treat? Is it going to be when you're 20? Or actually, there's no sign of the disease, you don't want to do it so young, rather do it 30, 40. So when is the time frame? And so rather than guessing, which would be unreasonable, costly, and maybe have a bad effect. What Michael is doing in his study, and is starting to hint at some interesting data, is he's taking these families, they don't have to necessarily know that they carry the mutation or not, and he's looking at different markers, brain markers, blood markers, urine markers, to see is there a clue at a particular point that something is going up, and that's the point that we would be treating. All unknowns at the moment, but that's exactly the way the research is thinking. Thank you for your good questions.
Yeah, it's hard enough getting patients into clinical trials now. By using biomarkers to appropriately include and exclude, how are you going to get enough patients together to power a trial and come up with meaningful results?
It's a good question, and I think there are many answers to that. The question of recruitment in general, which I'll leave to maybe the clinical discussions. The thing is that if you have a really good biomarker, you don't need to recruit so many patients. Take, for example, again, the best one that we have is blood pressure. If you're going to go into a trial and you have high blood pressure, you can quickly measure that. If a compound works effectively at that, you don't need hundreds of people to prove that it's done that. The reason why we have to have such big trials is, one, that the disease is so variable and our measures are not as solid. They're not bad measures, they're better than not having any, but they're not enough to be definitive to say that that is going to make a difference. So if you've got good biomarkers, your trials can be smaller.
And that will be enough to make the FDA happy about the safety aspects?
Safety is a very different thing to seeing if there's an effect. So trials for safety, and again, I'm not that's a very good question to re-ask in the clinical session. I'm not the clinical expert, but safety and efficacy, the fact that it's working, are different things. And you don't need as big a study just for the safety part of it. You certainly need a bigger study for the therapeutic effect. Rick, did you want to maybe comment to those questions?
Thank you very much. Just wanted to first make a comment about the earlier question about stories being lost. And many of us agree that that is tragic. And within the National ALS Research Group, we've got an initiative underway to create a database where we hope we will be able to eventually put the records of every patient with ALS in the country. So we're trying to make all the medical record systems that we're all using now compatible with a centralized database, so that when we enter information about a patient in our clinic, at the end of the day, if the patient says it's okay, we would actually have that information transferred to a central research database, because we think that every patient in the country is important as far as what's happening to them, and we don't want any stories or data to ever be lost. So hopefully we can make that work. Lucy, the question I had for you, and it actually does relate a little bit to this other gentleman's question, is one of the ways to maybe make research studies more attractive is to get patients involved earlier on. And I wonder if the ALS Association would ever consider, first of all, putting a patient on their scientific advisory board to help them select maybe some of the projects that are not only most attractive scientifically, but most attractive to patients, and that might generate more enthusiasm for some of the studies. And second, would there ever be a way to get patients involved in the design of studies? So even further on, before they even get submitted to the advisory board, could we create a system where investigators who are designing clinical trials might be able to tap into a bank of interested and educated patients to help them think about things from the patient perspective, so that patients might be designed with less burden, trials might be designed with less burdens, for example. Sorry, my name is Rick Bedlack. I'm a neurologist at Duke University.
So, Rick, I think those are both very valuable comments. Certainly in our research, more basic research programs, although we don't have patient representatives, we do have oversight committees that involve consumer interest, but they certainly don't do the review process. But that's certainly something that is I know DOD operates like that, and we work with that now too. So that's something to think about. In terms of the clinical question, I would be happy to work with clinical colleagues to see how to do that. We fund very few clinical trials at the moment, and our recent attempt to improve the clinical trial design was to focus on the end of biomarkers. But I think it's certainly a very valuable comment, and I think it's something we can certainly work through ALSRG and our networks and NEALS to help build that practically and valuably. So absolutely, I think those are valuable points. You do really, if the people with the mics can guide me, because I can't see very much with the lights.
Can I ask? I'm Sonal Shah. I'm type 2 diabetic, and basically I disqualify for every clinical trial. Is there a connection or reflection of having diabetes and ALS, or diabetes comes in the way of research?
So I will ask you to hold that question, or perhaps Dr. Sukovitz or Jeremy have noted that, so that you can address it now. If it's brief, that's fine too. Just, Mayor, 1 moment for a mic.
Thank you, Mayor Tsukovich. I would say it's actually just the opposite, that we see diabetes as less common in people with ALS. It doesn't mean it doesn't happen, obviously, but it's not a risk factor for ALS.
And why is it an exclusion in a trial, Merit?
Oh, usually it's not an exclusion for a trial. It would only be an exclusion if that drug might worsen glucose control or have some interaction with diabetes. And that does happen with some drugs, but most of them it doesn't.
So maybe if you have more additional questions, you might like to address it to Merit in the break, so she can help you with that. Other questions? I know it's an enormous amount of information, and obviously if you have questions that come up later or you didn't understand something, don't hesitate to contact us. I think we might have finished a bit early, which is quite remarkable for me, but I think you'll probably welcome a slightly longer break and a chance to discuss. But I mentioned you several times, Brian. You're in the back. This is Brian, our genetics guru, and who's going to take your blood if you're willing. So find your way, have a nice tea, and back at 10:30, no later, please.